Welcome to my poster. My name is Shanghua and I'm poster of Gabriel Lab in the University of Texas Medical Branch. The title of my poster is the Interplay between Heaven, Spark, MGs, modulators of new resin and new ligand transsynaptic bridge. In the nervous system, neurons are connected to neuron circuits by synapses. Synapses permit neuron impulses to transfer from one neuron to another. There are lots of proteins involved in the synapse formation. For example, the adhesion molecules, neuracin located on the presynaptic membrane and the new ligands located on the postsynaptic membrane form a transsynaptic bridge and promotes synapse development. The neuracin and new ligand transsynaptic bridges is highly regulated. Can it be disrupted? Can it be promoted? Can astrocytes that surround the neuracin neurons also alter synaptic connection, and how? For example, at inhibitory synapse, new ligand 2 bound to neuracin, promoting inhibitory zone. However, MDG1, a third synaptic adhesion molecule, decreased inhibitory tones, and it was hypothesized to block neuracin and new ligand transsynaptic bridges. Indeed, our lab revealed the molecular mechanism by which MDGs, here in magenta, bound to the new ligand, this yellow dimer, blocking the recruitment of neuracin, in blue here. Heaven and spark are a multicellular protein secreted by astrocyte. Heaven can stimulate eccentric synapse formation by promoting adhesion between neuracin alpha and new ligand 1. Spark, in contrast, does not stimulate synaptogenesis, and it can blocks heaven. Heaven and spark are very similar, but heaven contains very long N-terminal region that is not found in spark. So how do they regulate neuracin transsynaptic bridges? Our structure of heaven and FS domain tandem shows that FS domain here in orange made up of an EGF-like repeats and causal subdomain, and the EC domain in pink housing two canonical helix, loop helix, EF hands, and each contains a calcium bonding site. Strikingly, the heaven FS domain on left looking very different than spark on the right, despite the high sequence identity. There are two heaven molecules in our structure, a copy A and a copy B, so we can compare these as well. They are similar in green and pink here on your left. Superposing heaven and spark with their FS domain here on your left reveal that heaven FS domain are quite similar to spark FS domain, unlike EC domain. Superposing them with their EC domain here on your right reveal that that FS domain in heaven swings away from EC domain about 40 degrees compared to spark. Furthermore, while the two EF hands are structurally conserved between heaven and spark, here top left, the region we call the variable helix in cyan and magenta are dramatically different, top right and here. FPSC and FPR experiment demonstrate that having bound to different new ligands here on left, and that it is the FS domain that carries a major bonding site for new ligand, bonding quite well, about 16 nanomolar here on the right. The new resin 1-alpha contains 6 LNS domains and 3 EGF-like repeats. We use a PLC to show that having FS domain bonds to new resin as well, and in a calcium-dependent manner. In particular, the heaven FS domain makes the new resin 1-alpha, L5, L6 appear to recruit new ligand in our PSC. Here, this increasing green line, while mixed with new resin L2, L3, it does not. Our data suggests that having FS domain alone is enough to form tripartite complex with new resin and new ligand. Given the spark antagonized having action somehow, we test whether spark could also bond to new ligand and new resin. And indeed, spark bound to the new ligand here on top and two new resin here below, in a very similar manner as we saw of heaven. This result suggests that heaven and spark can both bond to new resin and new ligand, and they may compete with each other. Suggests that heaven FS domain and MDA's molecular share an overlapping bonding site on the surface of new ligand too, putting them in a position to compete with each other. Thus, an elaborate protein network of proteins regulates new resin and new ligand trans-neck bridges. MDJ can negatively regulate them, shown on the left, while heaven and can positively regulate them, keep in check by competition with the spark, shown here in the right. And these positive and negative regulators of new resin and new ligand trans-neck bridges compete for overlapping bonding site. We are very interested in these proteins because they are implicated in neuropsychiatric disorders like autism spectrum disorders and schizophrenia disease. In summary, we review an elaborate network of proteins that regulate new resin and new ligand trans-neck bridges, and the heaven FS domain bound to both new resin and new ligand with a direct interaction. Heaven also bound to new resin and new ligand, despite big structural difference in the EC domain. So heaven and spark luckily compete for each other, but spark does not induce synaptogenesis because it lacks a long N-terminal region. Amazingly, these positive and negative regulars of new resin and new ligand trans-neck bridges share overlapping bonding site on new ligand. This project was complete in the Rudenko lab. I would like to thank all the members of Rudenko lab, and as well as Mark White and Louis Hossein at the Seeley Center for Structural Biology for their help and support. Thanks for your attention.